Hello, everyone. I'm Wolfgang Kopp from the Max Delbrück Center for Molecular Medicine in Berlin, and I'm presenting dimensionality reduction and data integration of single-cell ATAC-seq data using deep learning. Single-cell ATAC-seq is an assay that allows us to measure accessibility across the genome in individual cells. And this is important for us to know because accessibility has been associated to regulatory activity. Single-cell ATAC-seq data is notoriously sparse and high-dimensional, which means we can't work with the raw count matrices. We have to first do dimensionality reduction before we can continue interpreting the results. And so dimensionality reduction is critical because this influences very often other downstream analysis tasks. We have developed a novel strategy for doing dimensionality reduction using a variational auto-encoder. So our model takes as input raw ATAC-seq counts, feeds this through an encoder, after which we can extract a latent feature representation. And then the latent feature representation is decompressed using a decoder, which computes a reconstructed accessibility profile. To make this model work well in practice, we use a novel reconstruction loss based on a negative multinomial distribution, which we find works very well for the sparse nature of single-cell ATAC-seq counts. A second thing that we incorporate is a batch correction strategy based on batch-adversarial training. So during training, we can fit the model such that it extracts a latent feature representation that is as informative as possible with respect to the cell state and the biology of the data, but at the same time that it's as uninformative as possible with respect to predefined batch labels. We benchmarked our strategy comprehensively using a published benchmarking framework. So basically, we take ATAC-seq data, fit our model, and extract a latent feature representation. And then we can compare whether we can recapitulate known cell types or cell labels based on our latent feature representation. So there is a part of this is shown on this plot here for real data sets. So we use the adjusted mutual information to measure how well we can recapitulate known cell types. So the higher they score, the better. And you can see that with our model, BAVARIA, we can do better in this case than the other methods. And in general, for other analysis that we did also in the benchmark, we can see that we are obtaining as good or better results than the other methods. We can also have a look at the UMIP integration and interpret the accuracy of the model from there. So what's shown here is a particular data set, Poinrose et al. In 2018, where hematopoiesis was studied. And on the right, you can see the UMAP that was built on the latent reduced dimensionality of BAVARIA. And you can see that known cell types are indeed clustering in distinct territories in this plot. Below, we look at the section of the genome, and we want to look at the accessibility profiles across different cell types. And what we compare here is the pseudobulk tracks of the original data and the imputation of the model. And you can see that these are in very high agreement across different cell types, suggesting that our model is indeed able to capture subtle features about the cell identities. We can also do data integration with the model, and we demonstrate this on mouse brain cells that were assayed with different technologies. One dataset was obtained using 10x genomics, another one with combinatorial indexing, and we would like to integrate these two datasets simultaneously. So here is the results of this integration. If you don't account for batch correction, you can see that cells from the different technologies are clustering in distinct territories. They don't overlap. But if we use our batch adversarial training strategy, we can align the cells from these distinct technologies. And this can then be used to transfer cell labels, for example, from one data set to another one, if one of the data sets had already been annotated, for example. There are more details on the analysis and the model in our preprint, and the source code for the model is freely available on GitHub.